Hello everyone, my name is Rital Al-Halawani
and Al-Jud Al-Marri.
And today we have a really interesting scientific topic to tell you about, targeting glutaminase to enhance oxaliplatin efficiency of colon cancer treatment in adults.
And first, we're gonna start with our researcher's problem. For our researcher's problem is that colorectal cancer is one of the most common and deadly cancers in adults, with cases in Qatar rising by 90% since 2015. And the main medicine for it is oxaliplatin, but many cancer cells has been slowly becoming resistant to it, so the treatment stops working well. And our project asks a simple question: can we weaken colorectal cancer cells by blocking their energy source? So oxaliplatin can kill them better. And here, as you can see, there is a graph showing how cancer has been developing every year by 2% since 2015 till today, 2026. Moving on to the research's main objectives.
First, we want to understand the problem in patients.
Second, test the idea in the lab.
And finally, we're going to use computer tools to study the drug combination.
Next is importance of our research. If cancer cells become resistant to oxaliplatin, patients may not respond well to the treatment, and our work shows that blocking glutamin use in cancer cells can make oxaliplatin more powerful, reducing cell growth, colony formation, and migration in the lab. This suggests a new combination strategy using a metabolism targeting drug together with standard chemotherapy.
Ultimately, this work aligns with Qatar's national health priorities by providing a data-driven, metabolism-informed strategy for precision oncology, offering a path toward more durable responses and improved survival for more patients facing this global health burden.
And as you can see, there is a picture explaining everything that we're talking about.
Now, with the research's main idea. Our research addresses the critical challenge of oxaliplatin resistance in colorectal cancer cells by targeting the metabolic glutamine addiction, which are made out of the glutaminase enzymes that the colorectal cancer tumor cells use. Our study demonstrates a significant sensitization for the colorectal cancer cells to chemotherapy, decreasing the migration and the colony formation and increasing the DNA damage. After using public cancer data and bioinformatics, the project provides a data-driven blueprint for more precision-based combination therapies. This works directly on the SDG 3, Good Health and Well-being, which also aligns with the Qatar National Vision for 2030, which is building a local capacity in advanced biotechnological research and AI. And it also aligns with the third goal for the Sustainable Goals of Development in Qatar.
And our research focused on three main questions. For the first one, we have patient data question.
The second is the laboratory questions, cell culture questions.
Third, AI computer model question.
Now, moving on to the researcher's hypothesis. Our main hypothesis is, if we block glutaminase in colorectal cancer cells, then oxaliplatin will work better, leading to more cancer cell death, fewer colonies, and reduced cell migration compared with oxaliplatin alone. Here we have a picture if we want to know more information about our hypothesis. Here we have the researcher's terminology. These are all the definitions we focused on in our research. For the next step, we have the researcher's results. Firstly, we have the TCGA-COAD datasets, and we have multiple graphs and diagrams that show us the GLS2 level in colorectal tumors versus in normal tissues. Secondly, for the researcher's results, we have an analysis of the GLS2 enzyme. We have a comparison of the tumor and normal tissues. As we can see in all the graphs and diagrams, the tumor is really going down. These are graphs for the GLS2, glutaminase 2 enzyme, and they're all going down, which means that the GLS2 enzyme is really elevated in colorectal tumors more than the normal tissues.
Thirdly, we have the MTT test that shows how SW480 cell line has been surviving with the oxaliplatin and 968 together. And for the other graph, shows the same thing, but oxaliplatin with CB839. Fourth, we have an experiment that shows how SW480 cells over a 14-day period reacting to control and oxaliplatin and CB839 and 968 alone and all together.
Fifth, we have the wound healing assay, which is an experiment where we scratch the SW480 cells in the middle. This took us 48 hours. As we can see, we have a study or a graph which shows us 6 hours, 24 hours, and 48 hours. With control, Oxaliplatin, CB839, CB968, and then all together.
And here we have Western blot analysis. We have two Western blots. For the first one, with the Oxaliplatin 968, and the second, we have the Oxaliplatin CB839. As you can see, there's two pictures of Dr. Ijaz's team that showed us how the Western blotting works.
Seventh, and finally, we have the AI augmented bliss synergy modeling that illustrated how the GLS1 inhibition disturbs the metabolic flux and antioxidant defenses. And we have a couple of pictures with Dr. Ijaz's team that showed us how the heat map AI-generated bliss works. Okay, now with the research's conclusion, our study integrates the bioinformatics and the functional assays with both the enzymes GLS1 and GLS2,
and AI-driven synergy modeling.
And here we have a picture if you want to know more about our conclusion.
And here we have some pictures of our lab experiments in Sidra Hospital that we did with the team.
Over here we have all our recommendations. Finally, we would like to express our sincere gratitude and thankfulness for the opportunity to work with Sidra Medicine and Dr. Ijaz and his team.
Thank you.